होते हैं उनको मिनिमम से मिनिमम डोज आती है एंड हम लोअर एमयूज रिक्वायर्ड अचीव कर पाते हैं.
Thank you, Dr. Prachi. 1 minute, Dr. Prachi. You mentioned that the risk of spread in the contralateral breast while radiotherapy. But one question from my side that if you perform the complete mastectomy along with the lymph nodes that is en bloc section, then do you still require to do the radiotherapy or only chemotherapy will be sufficient?
No, there is required radiotherapy required because अगर हम radiotherapy नहीं करेंगे तो there is a recurrence, local regional recurrence for breast and the regional lymph nodes.
Okay, even after the removal of the lymph nodes. Okay, thank you. Nice presentation and worth for the most alarming cancer in particularly in women. Thank you, Dr. Now next presenter is Subhlakshmi, scholar, GRCC Government Dover College, Bikaner, and the topic is microwave assisted synthesis, spectral and biological properties of GD3 complex with imidazole.
Good morning, myself Subhlakshmi.
Please sum up in 5 minutes.
Myself Subhlakshmi. I am the research scholar of GCRC PG Department of Chemistry, Government Dover College, Bikaner. My research supervisor are Dr. S.N. Jatodia sir and Dr. Narendra Bojak sir. I am greatly thankful to Dr. R.K. Purohit sir for giving me an opportunity for oral presentation. My topic is microwave assisted synthesis, spectral and biological properties of gadolinium third complexes with imidazole. The importance and versatility of derivative of imidazole is well established in many forms of drugs. Potential use of lanthanide complex as diagnostic molecule have been reported in literature, but study on biological aspects of complexes of lanthanide related with bioactive species are lesser. The magnetic moment of gadolinium third complex shows slightly higher value, which originated due to low JZ separation, leading to thermal population of next higher energy J level and susceptibility due to first order Zeeman effect. For various peaks of gadolinium third intensity, Z offend T2, T4 and T6 and oscillator strength, symmetry, coordination, RMS deviation, sigma parameter have been computed using partial and multiple regression method. In the present study, the properties of lanthanide are mainly focused on the comparative absorption spectrometry and the corresponding change in oscillator strength of different 4S transition spectra. In understanding the characteristics of
your objectives, very brief material method, result and conclude.
Yes, sir.
You have only 2 to 3 minutes.
The saturated solution of ligand and metal gadolinium third ion 0.05 m were prepared in different solvents. These solvent are Triton, SDS and HTAV is used for preparing saturated solution of ligand and metal. Absorption spectra of these solution at room temperature in 1:3 ratio were recorded in UV visible double beam spectrophotometer. Theory, oscillator strength. The intensity of transition, which is measured in terms of oscillator strength, is given by equation 1, 2 and 3. Z offend parameter. The electrostatic and magnetic interactions between lanthanide ion and ligand are predominantly measured by 3 main parameters known as Z offend parameter, that is T2, T4 and T6. And value calculated of these parameters from 4, 5 and 6 equation. Interaction parameters. These interactions can be expressed in terms of Slater-Condon, Lande and Raka parameter. The Slater-Condon F2 orbit coupling constant may be evaluated by expressing energy as Taylor series expansion. That is F2, F4, F6 and zeta 4F. Symmetry parameter T2 by T6 suggests a common symmetry around the central ion. Coordination parameter T4 by T2 suggests common coordination environment around lanthanide ion. Both the parameter T4 by T6 and T4 by T2 give information regarding change in ligand environment around central ion. And this is a table first and observed and calculated value of oscillator strength. Conclusion. The present study is based on doped crystal phenomena. RMS radiation between T observed and T calculated value indicate the applicability of Judge-Feld theory. Oscillator strength of transition 3S4 to 3P2 is high value which exhibit hypersensitivity to change in ligand environment. T4 by T6 and T4 by T2 value indicate almost identical stereo environment. Value of T2 found negative for micellar medium, which show mixing of FD orbitals. Stability of complex is sufficient in micellar system. Micellar medium is found to be best as intensity of UV visible spectra are high in micellar medium than alcoholic medium. Thank you, Dr. Narendra Bojak sir. Thank you, Dr. S.N. Jatolia sir, and thank you, Chairperson sir. Thank you, Dr. Tiporai sir, and thank you all.
Dr. Subhalakshmi, and as you are a scholar, it was nice presentation, and keep on trying. Next presentation, Dr. Vinod Kumari, Assistant Professor of Botany, Government MS College, Bikaner, and she will be delivering a talk on anti-cancerous plants of Thar Desert. Ma'am, please, you have got 5 minutes, so please be to the point.
Good afternoon, everyone. Myself, Dr. Vinod Kumari, working as Assistant Professor in Government MS College, Bikaner. Today, I am delivering a lecture on anti-cancerous plants of Thar Desert. As we all know that the Thar Desert plants are rich in secondary metabolites, and these secondary metabolites have some medicinal value also. And in this presentation, we will discuss about them. Our first plant is Prosopis cineraria. It is commonly known as khejdi and belonging to family Myosaceae. The leaf, stem bark, and seeds are rich in secondary metabolites. It is a life-supporting, multi-purpose keystone tree species of Rajasthan desert, and it plays a very important role in the social and cultural life of desert people. This tree provides food in the form of pods for local people and fodder to animals, as well as fuel wood. And during the drought conditions, this tree is very valuable for the local people. It is not this tree not only tolerates the extreme climatic conditions of Thar Desert, but also has a plentiful foliage, bear, flowers, and fruit during the driest period of the year. The immature pods, which is known as sangri, are used as a vegetable by the local people. Now, the bioactive compounds which is present in this tree is the whole plant contains 4-hydroxybenzoic acid, methyl 4-hydroxycinnamate, methyl 2-methoxy-5-hydroxycinnamate. The seeds of this tree contain prosterin C, prosterin D, prosterin E, gallic acid, pituitary, and the seeds contain relatively large proportion of unsaturated fatty acid with linoleic acid and oleic acid. Various studies conducted on this tree, and results are here. The anti-tumor activity of the hydroalcoholic extract of the leaf, stem bark of Prosopis cineraria in Swiss albino mice was evaluated against allylic acid carcinoma tumor model. Hydroxycinamic acid and chloric acid derivatives have been reported to possess antioxidant properties, and they are believed to reduce the risk of stomach cancer by reducing the formation of carcinogenic nitrosamines. Perturitin has been reported significantly cytotoxic activity against in vivo lung carcinoma. And the next plant is Caligonum polygonates. Locally, we call it as a foam belonging to family Polygonaceae. And the floral buds of these plants are utilized as a food. We prepare raita from these floral buds. Bioactive compounds which is present in these plants are all the plant parts are rich source of phenolic compounds with major portions of gallic acid, catechin, vanillic, chlorogenic acid, epicatechin, turmeric acid, catechol, vanillic acid, epicatechin, and cyanogen acid. It was reported that the presence of different secondary metabolites in different parts of whole plant has a higher scavenging activity against 2,2-diphenyl pipryl hydrazide and 2,2-azino-bis ethyl benzoline 6-sulfonic acid and superoxides. The HCl extract from the Caligonum polygonates possesses quercetin, which shows significant cytotoxic activity against MCF7 cell lines. The third plant is Capparis decidua, and we commonly call it as CAD, belonging to family Capparidaceae. All the parts of this plant are utilized for its secondary metabolite extraction, and we particularly use this plant fruits of this plant in the form of vegetables and in pickles. The bioactive compounds of this plant, particularly stecahydrin, which is an alkaloid, and stecahydrin poses significant cytotoxic effect on prostate cancer cells and result in a considerable inhibition of expression of chemokine receptor. Anti-invasive and anti-metastatic nature of stecahydrin makes it a competent candidate for the development of anti-cancerous drug. The next plant is Momordica palsamina. We commonly called it as a jhar karela. It belongs to family Cucurbitaceae, and the plant part used for the secondary metabolite extraction is leaf, fruit, and seed. This plant is we commonly see this plant along the roadside. And the bioactive compound of this plant is the leaves, fruit, seed, and bark, which contain secondary metabolites. And the therapeutic agent which is present in this plant is Momordil. In a study, the leaf acetone extract of this plant was evaluated for its anti-cancerous effect on breast MCF7 cancer line. The next plant is Citrulus colocynthis, commonly known as Tumba, belonging to family Cucurbitaceae. And the bioactive compound present in this plant is cucurbitacin glucoside. It is estimated that in vitro cytotoxic activity of these two compounds against hematoma cell line and mice bearing tumor of allied ascites carcinoma. Both these compounds has a potent anti-tumor activity on cell line. The last plant is Leptidemia reticulata, commonly known as Jivanti. It belongs to family Asclepiadaceae, and the whole plant possesses very good amount of secondary metabolites, rich in secondary metabolites, particularly the alpha and beta amyrin, ferulic acid, luteolin, diosmetin, rutin, beta cytosterol, and stigmasterol. And the ethyl acetate extract of this plant showed significant free radical scavenging activity, which revealed the anti-carcinogenic potential of this plant. Thank you. In a conclusion, we can say that we should use the fruit, seeds, or leaves of this plant in as much as in our diet, so that we can also protect from this dreadful disease, cancer. Thank you.
Thank you, madam. I have a question. Are you working with these plants, or you have just summarized?
Sir, I am working on the phytochemical study. I am working on phytochemical studies of some of the desert plants.
Yes. Yes. So, in what form you are using in as a raw leaves, and or you are
in some of the plants I have
them?
yes. In our lab, we have extracted through with the help of exoxylate apparatus, we extracted these the secondary metabolites of this plant, and in our lab we test the presence or absence of secondary metabolites. We have only this much facility.
Thank you, ma'am. Thank you. And the last presentation in this session, Purvi Vyas. She is a scholar, MGSU Bikaner, and delivering a lecture on radiation biology. Ms. Purvi, you please sum up only 3 to 4 minutes.
Stop. Please stop. Thank you so much.
Okay. Thank you.
Please, I thank the Honorable Chairperson, Dr. Parunji Bishnoi, Professor Head, Dr. Sakar Palicha, and Madam Saha. Since our guest of honor, today's guest of honor, Professor Shankar Jakhat sir, he is here. Our principal sir is here. Honorable Minister Sri Gajan Singh Shekhawat, he is online at 1 p.m. The memento giving ceremony we will do after at 1:30. Thank you, sir. Please. I would request Dr. Sonu Shiva to proceed the function. Nice.